So in this final video, I'm going to talk about oxidative phosphorylation. So this is the synthesis of ATP, and this is catalyzed by complex 5, which is also known as proton translocating ATP synthase. So complex 5 of the electron transport complexes. So in this case, the free energy that has been released by electron transport has been converted into a proton gradient, and we saw that in previous videos. And this proton gradient is now utilized to form ATP by ATP synthase. We can refer to the proton motive force, and basically what this describes is it describes that proton gradient and the fact that there is now energy stored in that proton gradient. And the energy is stored because the protons want to move back down their concentration gradient into the mitochondrial matrix, or if we're talking about bacteria, back down their concentration gradient across the plasma membrane and into the cytoplasm. So it's both a proton gradient and a voltage gradient, because of course protons are charged particles. This entire process of ATP production by oxidative phosphorylation is described by the chemiosmotic theory. The chemiosmotic theory was described by Peter Mitchell in 1961, and on this slide I've got several of the observations that support this theory. At the time there were a couple of different theories as to how ATP was synthesized, but this is the one that basically has the most evidence in favor of it, and is the one that we now understand to be what's happening. So these following bullet points, these are observations supporting the chemiosmotic theory. The chemiosmotic theory being that build-up of a proton gradient across the membrane, and then the discharging of that gradient being utilized to form ATP. So in order for this to occur, there has to be an intact mitochondrial membrane. And this is true. If you destroy the mitochondrial membrane, then you destroy the production of ATP from the mitochondria. The membrane has to be impermeable to ions, to charged particles such as protons, but also to other small ions like chloride ions, etc. Electron transport results in the transport of protons out of intact mitochondria. And this can be measured. You can actually measure the pH difference between the inside and the outside of an actively respiring mitochondria. If you add compounds that increase the permeability of the inner mitochondrial membrane, that allow electron transport to continue but destroy the proton gradient, these should uncouple electron transport from ATP synthesis. In other words, the electrons continue to be passed, but because you have destroyed the proton gradient, then you should not be able to make ATP. And finally, if you increase the pH on the outside of the mitochondria, then this should increase the rate of ATP synthesis, because of course an increasing pH basically is adding protons to the outside of the mitochondria, and therefore you should have a greater proton motive force for the protons to go into the mitochondria and generate ATP. So all of these observations support the chemiosmotic theory. This slide shows the electron transport chain and the complexes, and we've basically seen this slide before, but there's a couple of additional things I want to mention. So on this slide you can see complexes 1, 2, 3, and 4. You can see that complexes 1, 3, and 4 are proton pumps. Complex 2, of course, is not a proton pump. That's where the electrons from FADH2 enter the electron transport chain. You can also see coenzyme Q as being a mobile carrier that can move between complexes 1 and 2 and a complex 3. And you can also see cytochrome C, which is that peripheral membrane protein that can go along the outside edge of the membrane, transporting one electron from complex 3 to complex 4. We can then see complex 5, the ATP synthase, allowing the protons to come through the membrane and harnessing that energy in order to add a phosphate to ADP to produce AT. Now the other thing that you can see on this slide is this actually indicates where the genes for the various electron transport proteins are located. And if you recall, mitochondria have a small piece of DNA known as mitochondrial DNA. The mitochondrial DNA has 37 genes in total, and what we can see is that many of the genes required for the proteins of electron transport and for ATP synthesis are located on the mitochondrial DNA. So for instance, if we look at the subunits of complex 1, it's a very involved enzyme complex. In total there are 47 subunits, so this thing is huge. But 7 of those subunits, the genes for 7 of those subunits are located on the mitochondrial DNA. 40 of them are located on the nuclear DNA. So as you look down this slide, you can see that we have 13 of the subunits seen within electron transport and ATP synthesis are found on the mitochondrial DNA. And so that's 13 genes out of a total of 37 genes. So obviously a large number of mitochondrial genes are involved with electron transport and ATP synthesis. So a little bit about the proton pumping. We know that in an actively respiring mitochondria, the pH inside the mitochondrial matrix is about 0.75 units higher than the outside. So in other words, there is quite a gradient across the inner mitochondrial membrane. If we look at the delta G for proton transport out of the matrix, it's about 21.5 kilojoules per mole. And in fact, under physiological conditions, so not the standard conditions that we've looked at before, but under the physiological conditions, ATP has a free energy of between 40 and 50 kilojoules per mole. So in other words, it would seem that we need about 3 protons pumped in order to synthesize an ATP. For NADH, we know that one NADH supplies sufficient reducing power to pump sufficient protons to allow the production of 2.5 molecules of ATP. So we refer to the P to O ratio, which is the number of phosphorylations per oxygen reduced. We refer to this PO ratio as being 2.5. because basically 1 mole of NADH produces 2.5 moles of ATP. Of course, the PO ratio for FADH2 would be 1.5. We also have an inhibitor that blocks the flow of protons through ATP synthase, and that is oligomycin. And oligomycin is produced by Streptomyces and is an antibiotic. And so this will actually prevent the flow of protons and thus prevent the production of ATP. So if we look at complex 5, ATP synthase, we can think of it being composed of 2 major structures, but each of those major structures has many subunits. And so we usually think of it in terms of the F0 and the F1 regions of ATP synthase. And the F0 is the transmembrane region of the protein. So this is the bit of the complex that goes through the membrane. So this is either going to be the inner mitochondrial membrane if we're talking about eukaryotes, or if we're talking about prokaryotes, this would be the plasma membrane. And this is the region that forms the proton channel. So this is the region that the protons flow through on their way back down their concentration gradient. The second major domain is the F1 domain, and this is a water-soluble peripheral membrane protein that pokes out either into the mitochondrial matrix in eukaryotes or into the cytoplasm if we're talking about bacteria. Now each of these regions, the F1 and the F0, are composed of multiple different subunits, and we can see that on this slide here. And so we can think of the channel as being embedded in the membrane, and then the actual ATP synthesis region of the complex poking out into the matrix or into the cytoplasm. It is possible to dissociate the F1 and the F0 by using something such as urea, which will actually break those two portions of the protein apart. So let's look a little closer at the F1. So the F1 region is the domain that is sticking into the matrix or into the cytoplasm, and this has a fairly complicated structure. It's composed of 9 different subunits. The subunit composition is alpha 3, beta 3, gamma, delta, epsilon. So in other words, there are 5 different subunits, but there are 3 alpha subunits and 3 beta subunits. The alpha and beta subunits are arranged sort of like the segments of an orange, and on this slide here are shown in the red and the sort of the orange color, and they are alternating. And so we would see an alpha, a beta, an alpha, a beta, an alpha, beta as we went around the structure. The beta subunit is actually the ATP synthesis site. We can see the delta subunit is actually involved in binding the F1 to the F0 domain. If we look at the F0 region, so this is the region that goes through the membrane, this has a complicated subunit composition as well. This is made up of A, B, and C subunits, and I want you to look at the C subunits. These form a ring structure in the membrane, and in fact if we look at the figure below, you can see how each of the C subunits, and they can vary in number, there can be between 9 or 12 of these C subunits, it just depends on species, but each one of these C subunits is composed of 2 alpha helices going through the membrane. And so you can see these shown below, where one C subunit is shown in blue and one is shown in yellow, forming a ring structure that goes through the membrane. You can also see that the A subunit is actually the proton channel, and on this figure you can see the protons being shown going through the A subunit. And the B subunit, or the B subunits, form what we call the stator, which is basically the bit that connects the F0 and the F1 protein and actually holds the protein stable in the membrane, because those C subunits are going to move around in the membrane. We know that conformational changes in the F1 domain are powered by proton translocation. Remember, proton translocation means those protons flowing back into the mitochondrial matrix. And we can think of ATP synthase as acting like a 3-stroke engine with 3 chemically identical but conformationally different alpha-beta subunit pairs. So there are 3 alpha-beta subunit pairs, and at any one point in time they're going to be in 3 different possible conformations. We refer to these conformations as the L state, the T state, and the O state, and each one of these subunits is going to rotate through these 3 states, so at any one time each alpha-beta subunit pair represents one of these states. So in the L state we have ADP and inorganic phosphate loosely bound, and you can see this in the figure below. In the T state, the ADP and the PI are now tightly bound, and in fact that is where the energy from the proton translocation converts the ADP and the PI into ATP. And in the O state, the alpha-beta subunit is open and there is no ATP or ADP bound. So basically the 3-stroke engine means that at any point in time, one of the subunit pairs is in the L state, one is in the T state, and one is in the O state. And they just rotate through these 3 states as the protons pass through the proton channel, harnessing that free energy of movement, harnessing that kinetic energy to turn it into chemical energy in the form of ATP. A couple of other energetic considerations that we have to look at. So we're going to be producing ATP inside the mitochondrial matrix, and that ATP has to be exported in order for it to be used elsewhere within the cell and within the organism. And so ATP has a charge of 4- on it, and so that is going to be leaving the mitochondria. And ADP with a charge of 3- is going to be coming into the mitochondrial matrix, so it can be converted into ATP. Well, ATP is going to leave the mitochondria down its concentration gradient because it's being produced in the matrix, so there's going to be a higher concentration inside than outside, so it will want to move down its gradient. Also, it's going to want to move to the outside of the mitochondria because the outside of the mitochondria has all of those protons, and so it is more positively charged. So something that has a net more negative charge is more likely to move across the membrane towards the positive charges. So the ATP is going to be pulled out by the positive charges of the protons on the outside. This occurs through the ATP-ADP translocase antiporter. So ATP is exiting and ATP is coming in. We can also see some other transporters that are required in order for this to work. We know pyruvate has to enter the mitochondria so it can be used in the TCA cycle. And pyruvate enters through a proton symporter. So when pyruvate comes in, a proton is also coming in. So that is actually acting somewhat to destroy the proton gradient that is being built up by the electron transport. And we can also see when phosphate comes in, it comes in with a proton. So we have a couple of things that we have to consider here as far as the transport of different molecules across the membrane and how those are actually going to ultimately affect the proton gradient that is established through electron transport. Every time you allow a proton in via one of these other channels, you are of course reducing the effect of the proton gradient that has been built up. Every time you allow a negative charge out, you're going to again reduce the effect of having the negative charge inside to pull the proton back in. So all of these things do actually affect the energetics of the system. About 25% of the energy yielded from electron transfer is consumed to regenerate the electron potential that is affected by the transport of ATP and ADP. So changing those charges has the effect of decreasing the proton gradient to about 25%. So this is not an inconsiderable loss of energy. It is possible to uncouple electron transport from ATP synthesis. So normally electron transport and oxidative phosphorylation are very tightly coupled. In other words, the proton gradient that is established is only allowed to re-equilibrate by passing through the channel of ATP synthase. However, uncoupling is a process of allowing those protons across the membrane in a way other than through ATP synthase. So this effectively breaks down the proton gradient without producing energy. The kind of drugs that can do this are generally these weak lipophilic acids. So in other words, they're weak acids. They can donate protons depending upon the pH of the environment, but they are lipophilic. In other words, they are mostly fairly hydrophobic, so they can get across the membranes. So if we look at the top one, the 2,4-dinitrophenol, usually abbreviated DNP, you can see the acidic proton on the phenol group, and basically as this passes through the inner mitochondrial membrane, it's going to reach an environment inside the mitochondria where the pH is higher, and so it can carry a proton across the membrane. When it gets into the mitochondria and the pH is higher, now the pH is above the pK of that proton is released. So effectively what the dinitrophenol has done is it has released a proton, it has brought a proton into the mitochondria without it passing through ATP synthase. So it has uncoupled the proton gradient from the production of ATP synthase. The bottom molecule, the FCCP, behaves in exactly the same way. In the 1950s, dinitrophenol was used as a diet drug. It was very effective because basically if you took the dinitrophenol, then you would uncouple the proton gradient, you would not produce ATP or not produce as much ATP, and so you would certainly lose weight. It did have a major side effect, and that side effect was death, which is not a very good side effect when you're trying to lose weight. And the reason for that is because when you uncouple the electron transport from ATP synthesis, the energy has to go somewhere. And where does the energy go if it doesn't go into ATP? It produces heat. So effectively what happened when people took dinitrophenol is they would actually overheat to the point of it being dangerous. So I don't suggest you try dinitrophenol if you want to lose weight, though you do lose weight if you die. Other situations where uncoupling is utilized to produce this heat is in brown fat tissue. So brown fat tissue, which is found in newborns, so newborns that lack fur, which would include us, and also in animals that hibernate. And this is how hibernating animals actually can keep producing heat as they're hibernating. So here we have this hibernating bear, and female bears give birth to their cubs while they're actually hibernating, and so the female will essentially be asleep and give birth to the cubs and be feeding the cubs for a couple of months before she comes out of her den. So the cells in this brown fat tissue contain a protein known as thermogenin. This is also known as uncoupling protein, or UCP. And essentially what thermogenin does is it does what those chemical uncouplers do. It forms a channel in the mitochondrial membrane that will open, and it's under hormonal control. So under hormonal control the channel opens and allows the protons to move back into the mitochondria without producing ATP. The energy from this proton flow, instead of producing ATP, produces heat. And so this is how the brown fat enables these animals to stay warm whilst they're hibernating. Finally, the control of oxidative phosphorylation. This is of course very important. The total needs of a human body for ATP in one day are over 2,000 times more ATP than is present at any one given time. A typical adult requires about 200 moles of ATP, but only stores about 0.1 mole of ATP at any one time. If ATP production stops, then the body will very, very quickly run out of energy. The amount of ATP required varies greatly depending upon activity. From sleep to activity, it may have to increase up to 100-fold, so that's a big increase that has to occur. Most of the control is occurring at the control of the production of NADH or FADH2. So in other words, by up or down regulating glycolysis in the TCA cycle. And we have to remember that the TCA cycle is not going to run if oxygen is not present. So under anaerobic conditions, glycolysis is going to have to be upregulated greatly in order to meet the needs for ATP. And glycolysis can do this because the enzymes of glycolysis are present in very large amounts within the cytoplasm of cells. So it's pretty easy to upregulate glycolysis to pick up the slack if oxygen is not available. The issue being that glycolysis cannot continue at those high levels for long periods of time. Because of the production of lactic acid, the pH is going to decrease within the cells, and several of the enzymes of glycolysis, especially the phosphofructokinase, are quite sensitive to changes in pH. So if the pH decreases too much, the phosphofructokinase, which of course is the main control enzyme is actually going to decrease its activity and stop working. But we can see great changes in upregulation and downregulation of the glycolytic enzymes. Okay, that's what I want to say. Thank you.